Greetings all, I'm Farizal Fahmida from the Department of Microbiology. I'm here to present the topic on tobacco mosaic virus. So the disease name is tobacco mosaic disease and its causative agent is tobacco mosaic virus. Host plants are from the Solanaceae family such as tobacco, tomato, pepper and other related crops. It can also infect certain ornamental and weeds which can act as a virus reservoir. So the morphology of this organism is it is a rod-shaped virus with a helical symmetry with the size of 300 nanometer in length and 18 nanometer in diameter. It is a positive sense single-stranded RNA virus encapsulated within a rigid protein coat called as capsid, and it is made up of 2130 protein subunits. TMV lacks a lipid envelope which contributes to its extreme durability and resistant to environmental conditions such as heat and desiccation. So, the history of diseases, it is one of the first virus ever discovered and extensively studied for over a century. In the late 1800s, Adolf Meyer, a German chemist, began investigating the mosaic disease in tobacco plants. He found that the disease could be transmitted by rubbing plants from infected plants onto healthy ones, similar to bacterial infection. However, Mayer didn't fully grasp the nature of infectious agent, believing it to be a soluble toxin. Then the Russian microbiologist Dmitry Ivanovsky between 1887 and 1890 demonstrated that the infectious agent could pass through filters that retained the bacteria. Then, the Dutch microbiologist Martinus Beijerinck independently replicated Ivanovsky's work and concluded that the agent was not a bacterium but a contagium vivum fluidum. This marked the beginning of the concept of viruses. Then, the characteristics of this disease is TMV is a positive sense single-strand RNA virus which is highly stable and capable of surviving in dried plant material for years. In 1898, Martinus Beijerinck identified the infectious agent as Contagium vivum fluidum, which is later termed as virus. It is prevalent worldwide in both field and greenhouse conditions. TMV does not require a biological vector. Instead, it spreads through mechanical means such as human handling, tools, and contaminated materials. Due to its high environmental stability, the virus remains infectious in dried plant material for years. This ability to survive and infect across seasons without a host make TMV a serious concern for crop production. So, the TMV enters plant through wounds. Infected sap from a diseased plant can contaminate hands, tools, soils, or equipment, which then spreads the virus to healthy plants. Seed transmission may occur if seeds coat are contaminated. On pathogenesis, once TMV enters the plant through wounds or natural openings, it begins by uncoating its RNA genome inside the cytoplasm of host cell. This RNA is then translated by the host ribosome to produce viral replication protein. These proteins facilitates the synthesis of complementary RNA strands which serve as a template for producing more viral RNA. TMV replicates rapidly and captures the plant's biosynthetic pathways by assembling new virions within the cytoplasm. The virus moves from cell to cell via plasmodesmata and systematically through the phloem to distant plant parts. During pathogenesis, TMV disrupts the normal cellular metabolism and photosynthesis, resulting in the appearance of mosaic pattern, chlorosis, and other typical disease symptoms. Symptoms of TMV infection include light and dark green mottling of leaves, leaf curling, stunted growth, distortion of leaf plates, and necrotic spots. There is no known chemical treatment for TMV once the plant is infected. Management of the disease focuses on prevention and cultural practices to limit the spread. Control measures include using resistant cultivars, disinfecting tools and equipment, avoiding tobacco handling near plants, and removing infected plants properly, practicing crop rotation to prevent virus buildup in the soil. Preventive measures involve maintaining strict field hygiene, educating workers on sanitation protocols, avoiding the use of infected plant material, applying disinfectants like sodium hypochlorite or trisodium phosphate to tools and surfaces. Using virus-free seed and seedlings can prevent the spread of TB. Thank you for watching.